Hey, beer appreciators, Swaddy Krab. Welcome to Beer Time Story, the platform where beer and brewing knowledge is shared. For today's session, we will have some good burps about role of oxygen in yeast daily life. Nowadays, it has become more and more common knowledge that oxygen is one of the essential nutrition for yeast, which we have already touched on it, in the yeast essential nutrition, they are what they eat episode with Nate from S Government Lab. Well, to be honest, we just scratched the surface of this matter as there were a lot more, to learn about role of oxygen for the yeast. And it is worth having a dedicated session for this topic, which is why we are here today. Yes, in this episode, we will not only further explore the role of oxygen for yeast, but also try to uncover what oxygen is to them. How and where do they use oxygen in their daily life, as well as discuss the possibilities of substituting oxygen with other supplements. To go through this set of questions, I will need a big help from a good hand, our guest speaker, so to speak, who happens to not only spend, about half a decade researching about the stress response of yeast, but also continuously, support the brewing industry with his yeast-related products and knowledge for over a decade. And here is a snapshot of his background. After receiving the PhD in microbiology and molecular genetics, instead of going straight down this path, he spiced things up by shifting gear and went to law school in 2006. His plan was to become a patent attorney for the science industry, which he actually did for about 4 years, representing the biotechnology group in Chicago from 2009 to 2013. Now, during Christmas of 2012, while he was having lunch and chatting with his friend, who was working on opening a brewery in that local area, their conversation came over the topic about yeast and how his friend found, how financial challenging it was to order liquid yeast and have it shipped overnight to the brewery because there was no local yeast lab around Chicago area at that time. That was a light bulb moment for him. It clicked that second, and that day he went home with an idea to start a yeast lab. Now, about 6 months later, in July 2013, the yeast lab he found opened the door to the brewing world under the name Omega Yeast, with the aim to provide, high quality pitch ready liquid yeast for both pro and home brewer. Fast forward to the present day, some of the yeast strains that his lab are widely used in the industries and may ring your bells are West Coast 1, British Ale 5, Lutherakovike, Star Party, and Daybreak 5. In addition, he also launched an online educational platform that shared their groundbreaking research, discussed the experiment, as well as provide insight, information, and advice about yeast and fermentation called Top Crop, which is a very valuable source of knowledge. So please welcome our guest speaker, Lance Chandler.
Thank you. That was a great introduction. You told it better than I could.
Oh my God. Thanks, man. Hey, welcome to the platform. Appreciate that you joining us today.
Yeah, thanks for having me.
Okay, cool. So how are things over there, Lance, in general?
Yeah, I mean, good. We're heading into spring, so things are warming up because the Chicago winters can be pretty unpleasant. So it's always really good for everybody's mood when we start shifting towards spring, when everything turns green again and weather warms up. And it's one of the best times of the year to be in Chicago. So,
Nice.
Good from that perspective. Yep.
Okay, well, so I want to lightly touch on, the Omega yeast a little bit. Can you tell us or can you give us some of the sneak peek of something interesting, the project or research that you guys currently cooking in your lab?
So we'll be announcing a couple of new strains at so the Craft Brewers Conference is coming up, the biggest trade show in the US relating to craft beer. So that's in Indianapolis in a couple of weeks. So we'll be talking about some new strains we have coming up, including some acid producing strains. So basically some brewing strains that can make sour beer all in one, so you don't have to use bacteria. that, and some diastatic strains. So basically strains where we've knocked out the STA1 gene, so you can make saisons without having to use what can be a risk in the brewery, which is a diastatic strain. So those are the announcements we have impending. Probably early next week we'll be talking about those, but I guess there's a little preview.
That's cool, man. All right, and I would like to warm us up a little bit before we dive down today's topic. And I realized that apart from those traditional brewing yeast strain that you offer, there are also those that utilize an innovative engineering method in the making from your lab, which deliver unique properties to the yeast, like the thiolize or DKO or lumina. Lance, how did you and your team come up with the idea of creating these characters? And can you briefly touch about the technology that drive this creation?
Sure. I mean, the underlying technology that we use to change any of these strains is called CRISPR. So it's basically a DNA editing tool, so it can be used in lots of organisms, yeast, and I mean, it's starting to be used to do human medical treatment. So there have been people that have had their like sickle cell anemia fixed with CRISPR. So it's a groundbreaking technology that really only came about. I think it was like 2012. It's not an old technology, but it was groundbreaking with the precision and ease in which you can make changes to DNA. So that's the tool we use. But some of the ways in which we've applied it would be, like you mentioned, the thiolized yeast. So we've got a couple of versions of that, but the idea is that it provides an enzyme to the yeast that can take precursor forms of a compound called a thiol, which there are lots of different thiols, but in beer, the ones we're worried about are ones that smell like tropical fruit, like passion fruit. So there happens to be a lot of these precursor forms of that thiol in malt and hops. So, and in that form, we can't smell them. But if yeast have this enzyme, they can release that compound in a form that we can smell. So, and it smells a lot like a New Zealand Sauvignon Blanc, if you've ever had something like that. I mean, it's really pungent. So that's one example. In another example, we've knocked out the phenolic gene. So we've taken like the famous Hefeweizen strain, which it's very, banana forward. But it also makes phenolics, which give you that kind of clovey smell. And we basically broke the gene that makes that clovey smell. So in the same way that lager yeast already have a broken version of that gene, or American ale yeast already have a broken version of that gene. So broke that gene in that so that you only get the banana, you don't get the clove. And then, the Daybreak 5 uses, we discovered what we call the haze gene. So the gene that's responsible for making hazy IPA when you dry hop with certain strains. So certain strains have a version of that gene that make it very, very hazy when you dry hop. And so we broke that gene in that hazy strain. So now you've got more of a strain that's kind of similar to Chico in that it's reliably making a clear beer, but it has the soft mouthfeel and ester quality of a hazy IPA strain.
Gotcha.
So we use that tool in different ways to break genes, to add genes, to tweak genes.
That's cool. So that the CRISPR-Cas9 genetic engineering tool is very groundbreaking, and the way you embrace them and come up with an idea to tackle different problems in terms of fermentations and yeast performance is also fascinating as well. So I myself found that with this kind of new offering for the yeast selection, it also allows brewers to be more meticulous, or have more tools to play around as well. So thanks for that.
Oh, sure, yeah, and that's definitely the way we look at it. We want to get tools out there and then kind of leave it in brewers' hands to be imaginative with it, and they come up with ways to use it that we weren't thinking about. So it's craft brewers are very creative people.
Yeah, sometimes a bit crazy too, but in a good way.
not all the experiments work, but that's the nature of science.
Right. Okay, that's cool. That's cool. I think we are ready. It's the right time to talk about the oxygen and yeast. So to roll the dice on, or just to roll things up, I believe that it is best to look at the big picture first. For our case, Lance, let's talk about the relationship between yeast and oxygen, as a big picture. So what is oxygen to yeast, and when yeast uptake oxygen, does it always mean that they respire, like breathe air, like what we do?
It does not. So oxygen means a lot of things to yeast, and they can use it in that way, but they don't always. So yeast is a very unique organism in that if there's a lot of sugar around, high amounts of sugar, even if there is oxygen around, it doesn't respire. And that's like in all of the kingdoms of life, that's an unusual thing to do, because the use of oxygen to completely break down sugar to respire, in other words, is very energy producing. So it's an efficient way to produce a lot of energy, and yeast don't do that necessarily. If there's lots of sugar, they preferentially ferment. In other words, they don't use the oxygen to respire, instead breaking the sugar down into ethanol and carbon dioxide. So, but if there's a very small amount of sugar, they will respire. So they will use it in the same way that we use oxygen, and they'll use it very efficiently. And that's what the lack of respiration in the presence of oxygen, high amounts of sugar, is called the Crabtree effect. And it's very unique to yeast, to the extent where we're not entirely sure why they do that, because it seems like you shouldn't. It's just not an efficient use of sugar. And one of the theories behind that I find interesting is that it's kind of a competitive thing. So they can produce high amounts of ethanol very, very quickly when they ferment, and that inhibits the growth of all the microbes around them. So some people think it's a competitive thing. And then if there still happens to be oxygen around, they can actually respire ethanol. So they can consume the ethanol themselves and respire it and make energy out of it. So it's not like it's not all is not lost if they produce. I mean, that's not obviously what's happening when we drink beer, but yeast doesn't exist to make beer, even though that's how we've kind of commandeered it. But so yeah, I don't know.
But it seems like they are greedy. They want to preserve all of those food for themselves, and it is pretty interesting to hear that. So like oxygen is vital for them in terms of it has something to do with the way they extract energy. If I have to put it into a more simpler word, but even in the environment that is rich in oxygen, but if you have a certain amount of say sugar, they still kind of switch their function, which is very interesting. That's called Crabtree effect, right?
Right. Yep. And as I said, in the scheme of nature, it's a very unusual thing, just because it's not an efficient use of that sugar. But a lot of like dry yeast manufacturing company, for example, they actually, when they're growing their yeast, they do have them respire. So they'll slowly feed small amounts of sugar so they don't trigger that Crabtree effect, because they can produce far more yeast mass out of a smaller amount of sugar if they respire, because it is a more efficient use of that sugar. But in order to do it, you have to closely monitor the amount of sugar they're getting and keep it below that level that triggers the Crabtree effect. So it's a very difficult thing to get yeast to avoid.
Right.
As a practical matter, any home brewer they're always triggering the Crabtree effect. Like even if you make a starter at home, it's got the lid off, so it's getting oxygen, it's still fermenting. Even though there's oxygen coming in there, it's still fermenting.
Well, it's very interesting to hear, the way dry yeast manufacturers kind of really tailor the precise process to not trigger the Crabtree effect.
Yeah, and just generate the most biomass from the least amount of sugar.
Got you. So moving forward, next thing is that we briefly touched about the word respiration, fermentations, and breaking downs of sugar. I understand that, like what you mentioned, yeast is a very resilient living organism, right? They can live in various kind of environment, including those that are hostile too, and they have their own surviving mechanism, for those scenarios as well. And one of the key thing is to extract energy from breaking down food, like us. We eat food to fuel our body,
Mhm.
and from a closer look, there are two ways that they can do that. One heavily relies on the presence of oxygen, and the other is on the opposite end of the road. So I think let's talk about this matter first, and we will see how oxygen come into play, right? Um, let's start with the oxygen-rich environment, the so-called aerobic respiration. So, Lance, what is it, and where does oxygen fit into this picture?
So yeah, respiration basically, well, as you're showing it there, yeast or any organism breaks down sugar completely into carbon dioxide and water. And they do that through a series of reactions through glycolysis through the TCA cycle. And they generate energy through that. And the form of energy they get from that is called ATP. It's just a chemical form of energy. So It's a very efficient process that basically extracts energy out of the oxygen and the sugar molecule in the most efficient way it can, and generates lots of energy. But it requires the presence of oxygen to do that.
So basically, when they eat sugar, say the simplest form is glucose, right? In that whole process, glycolysis, TCA, and electron transporting stuff, oxygen is involved in the equation, and at the end of the game, they get a lot amount of energy. I think that what people are mentioning now is around 30 to 36 unit, right, of ADP. So that seems to be quite a hefty amount of energy that they can produce per 1 unit of glucose. Okay,
yeah,
so
and we'll see how different that is from
Yeah.
when we get to fermentation, which is not nearly as efficient.
True. Okay, so moving on to the next one, which is in the absence of oxygen, the yeast do the things called anaerobic respiration, and for brewer, we may familiar it as alcoholic fermentation. So what happened here, Lance?
Yeah, so the first part is actually quite similar. Glycolysis is pretty much the same. But at the end of that, you yield two pyruvate molecules, and that gets broken down into ethanol and carbon dioxide in yeast. For lactic acid bacteria, it's breaking it down into lactic acid. And again, it's a much less efficient process. So we get 2 ATP molecules per sugar molecule, and 2 molecules of ethanol and 2 molecules of CO2. So it's just a less complete breakdown of the sugar molecule.
So this is what we put them through daily, right?
Yeah, we use this process to make beer, to make wine, and the beginning process of distilled spirits. It's this is what's going on.
Right. So in comparison with aerobic respiration, 30 somewhat unit of ATP, for these fermentations, they have only 2, which is really insufficient. Insufficient ways or ineffective way for them to produce. But yet their nature tends to trigger this path. Right, when there are not enough oxygen and with a high amount of food.
Right, and that's where they differ from, if you, like something like E. coli, where if you gave them lots of sugar and lots of oxygen, they would respire. But yeast will still ferment even when that oxygen is present. So they're not using it for this process, but they still need it for other things.
Gotcha. So this is the main mechanism of how they extract energy in 2 different ways, with which oxygen and which poor oxygen environment. Well, resilient kind of organism. Okay. I mean, we see the difference of the way they break down the food and extract energy, and how oxygen comes into play. But I think looks like something is still missing here. When yeast undergoes alcoholic fermentation, it doesn't require oxygen. But at the beginning of this session, we said that oxygen is, the vital essential nutrients for the yeast. So for this case, Lance, what does the yeast use oxygen for then? Yeah, let's just talk about this subject.
Yeah, so there's basically two other requirements for oxygen, and that is to make unsaturated fatty acids, which I think we can probably just call UFAs because that gets unwieldy to say unsaturated fatty acids over and over again.
All the time,
So UFAs and ergosterol. And both of those things are components of the plasma membrane. So if we're looking at a yeast cell, well, there you go. It's right in front of us. So the plasma membrane is basically what separates the yeast cell from the environment. So it defines the outside of the yeast cell. And it's basically a bilayer of fat. So the hydrophobic ends of these fat molecules interact with each other, and the charged ends are on the outside and inside of the yeast cell. So they need unsaturated fatty acids to have the right kind of flexibility and the right porosity for that cell membrane. And ergosterol is kind of it's a different type of molecule. It's actually more related to like cholesterol. So we have cholesterol in our cells, and fungi like yeast have ergosterol. They look pretty similar at the molecular level, but they're different. And it's the same thing. They need it for fluidity of the membrane, just so it has the right integrity to, house the proteins that span that and things like that. So oxygen is in both of these molecules, and molecular oxygen, O2, is needed for the yeast to produce unsaturated fatty acids and ergosterol. So this is not an energy producing sort of mechanism like it is for respiration. It's just part of these molecules that are needed for a healthy cell.
Gotcha. And first thing is, can we talk about what could possibly happen if yeast does not have enough UFA and sterol resource? What could possibly go
wrong? Well, eventually they just stop growing, and that I don't honestly know if people have worked out exactly what happens to the cell and why it can't grow. I mean, it probably relates to it can't take up nutrients as efficiently, or the proteins that span the cell membrane aren't functioning properly if the fluidity of the membrane is not right. But at the end of the line, if they don't have unsaturated fatty acids or ergosterol, the cells can't grow. They just won't grow, reproduce.
Gotcha. And in terms of the cell membrane integrity, the permeability and fluidity, it seems like, if you have this I would say fat molecules in the membrane, it makes them becomes more pliable and more adaptable to the environment, right?
Right. I mean, I think I've heard the kind of analogy, like butter doesn't have as much it's got a lot of saturated fatty acids, so it's a very ordered compound, and when it stacks up on each other, it makes that's why it's solid at room temperature. Whereas unsaturated fatty acids don't stack up nicely, so they're fluid at room temperature. So we can kind of think of it that way. We need some amount of fluidity for that cell, to have that pliableness to allow proteins in it and to take up other molecules that it needs to grow. It can't be like butter, it's just solid at room temperature.
It's pretty weird to think that. I mean, like solid yeast cell, but yeah. And just for our audience, you may not aware of this, but yeast technically they don't have mouth, so they uptake food via their cell wall and cell membranes, right? They have ports and all the stuff to transport in and export things that they don't want. And I believe that part of the health or the integrity of the membrane play significant roles of how they regulate things in and out of the cell.
Right. Yeah. I mean, there are, like transporters. I mean, they have specific proteins that embed themselves in the membrane that bring sugar into the cell. They have, different transporters that bring amino acids into the cell. So yeah, they can't just let anything in, right? And they can't let anything out. So it's a very regulated process. So in order for all that to work, that cell membrane has to have the right mix of sterols and unsaturated fatty acids and saturated fatty acids. It's got to have just the Goldilocks zone to be just right to allow all these mechanisms to work properly.
Right. And probably one of the analogy that I can think of is probably when we have like a dry lips, during the cold winter, and we try to maybe we yawn too fast and the cracks break and we hurt our mouth so bad. It's probably maybe in the molecular level kind of happen if they run out of those resource of fats, possibly.
Sure. Yeah, that's fair.
Okay. And when you touch about if they are not sufficient amount of these fats, they will tend to stop reproduce, which is via budding, right? And when the yeast bud, they share the membrane with the daughter cell as well. So those reserve of fats and other stuff will have to be split.
Right. So if they're, and there were some experiments I talked about in the Top Crop article where, you can get, even if they don't have oxygen, they don't have fatty acids that you're feeding them or sterols that you're feeding them, they will still reproduce a few times. And the reason they're able to do that is because they are sharing those UFAs and the ergosterol with the daughter cells, but there's only so much to go around, so they can't do that perpetually.
Okay. So the last question for this subject is that so oxygen seems to be the very source of this UFA and sterols. Are there any other source of these fats that yeast can get from the natural brewing ingredients?
I mean, even barley supplies some amount of unsaturated fatty acids. So if you could actually feed yeast these things in the absence of oxygen, and they would be perfectly happy. So they don't have to make them from scratch. They're perfectly content being fed these things. But if they're not being fed those things, then they have to make them in order to grow. And in order to make these compounds, they have to have oxygen.
Gotcha. So if they can just, get the food or get the essential stuff without have to work, I think they're going to be happier.
Yeah. I mean, they don't have to waste the energy doing it. They don't care whether they have to make it or whether they can take it out of the environment, but they need it one way or the other.
Okay. Gotcha. Before we head to the next section, which is about the articles from the top crops, I would like to ask you, this question is probably, sidetrack the conversation a little bit, but it also related to the oxygen and the role with yeast. So say in the lag phase of fermentation, right? After we oxygenate the wort and pitch the yeast. So yeast use oxygen to synthesize unsaturated fatty acids and sterol. But my question is, does yeast also utilize oxygen to create energy via aerobic respiration?
I mean,
in the same time?
Possibly a little, but I mean, honestly, not significantly. Because like I said, you can be pumping oxygen in there, but if that sugar content is high, they're not going to do it to an appreciable level. And I mean, you can determine that by just measuring alcohol content. Like you can, at the end of that process, you're going to have ethanol, even though there was oxygen there that whole time, assuming something that you're pumping oxygen.
Right.
So that's just that power of that Crabtree effect. So, so I mean, they may be doing it, but it's to an insignificant level. I would say in the beer world, and when we're using yeast to make beer, the primary importance of oxygen is for the things we just talked about, for making sure they have unsaturated fatty acids, UFAs and ergosterol.
Got you. I mean, just to put things in my head to the right spot and try to clear up some haze a little bit. Even we put like a high amount of oxygen, it's still kind of tend to ignore the aerobic respiration form. But it may happen in an insignificant level that you mentioned.
Yeah. I mean, if it was happening to any significance, we'd see that in the lack of ethanol production, because ethanol is not a product of respiration.
That's water and CO2, right?
Yep, that's it. Yep.
Okay. That's not the thing that we look for.
Nope, they're not going to detect the water they're making amongst all of the other water there. Yeah.
All right, cool. Okay, I mean, so far we've learned about role of oxygen in yeast daily life. The importance of for the yeast in terms of energy production, as well as the cell membrane integrity. Now, for this part, let's talk about the supplementation of oxygen. We know that we can oxygenate the wort for the yeast to get oxygen, and there are also fat content that's available in the barley or the grain itself that yeast can uptake. But in the past, there were several research or study that look at the stuff that can be added to the wort to replace oxygen. I'm not really sure why they would want to do that, but it happened. And one of the supplements that was widely discussed was olive oil, like the one we use in the cooking process, which shows some interesting result. However, it may mislead at some point. And I understand that, Lance, you have taken a look into this theory and research quite a lot, and you probably have some thought about it. So let's talk about the article that you published in the Top Crop, olive oil versus oxygen. What is it all about?
Yeah, so this is, I mean, I guess you see it popped up in a lot of different places over the years. I think the first place we saw it appear was in something that was done at New Belgium. And I think the original theory behind it was if you could use olive oil, small, and we're talking very, very small amounts. It's not an amount that's going to make your beer taste like olive oil. But if in theory you could use something like olive oil as a replacement for oxygen, then you're minimizing the staling effects that come from oxygen. As we know, like once you do that initial oxygenation when you're making beer, the brewer does everything in their power to avoid introducing oxygen from then on, because it becomes, a staling agent. So I think the original theory was, well, if we can, it's easier too. If you're not having to like inject oxygen into your wort as you're sending it to the fermenter, that's great. And you just do it through a little simple addition of olive oil. That's also good. So the theory was that you could just add this olive oil, and now the yeast doesn't have to use oxygen because we're providing the UFAs to them. And so that's kind of the original theory that New Belgium was kind of operating with. And a couple of people took that up later. New Belgium saw delays in completion of fermentation, so it did seem like it wasn't total replacement, because they didn't see as good performance from the yeast. And I think their chemical analysis of the beer suggested that it was slightly more estery than the control batch, but it wasn't too significant when it came to tasting panels. So I think them seeing a slowdown in fermentation was enough for them to say, well, this isn't going to work. But it also didn't show, it didn't destroy the beer. The beer was still perfectly fine. And that seemed to be about as far as they took it. A couple of other people took up the same thing. But all of these studies kind of ignored the fact that yeast need ergosterol too. Like UFAs are not ergosterol. They're not the same compounds. They don't do the same thing. But they both happen to need oxygen for the yeast to make them. So sure, it's not crazy to think that you can give or add a little bit of olive oil to your fermentation, and now the yeast don't have to make unset UFAs. That's reasonable and I think true. But it doesn't solve the second half of that equation, which is the need for ergosterol. So again, as we talked about earlier, you could supplement ergosterol, but there really isn't a readily available cheap supplement. It is something specific to fungi. Like fungi are the only things that make ergosterol. So there are, you know, scientific sources of it, but there aren't cheap sources of it. Olive oil and the amounts you use really cheap. But there is no cheap source of ergosterol. So, and, you know, everybody that looked at this, I think, kind of saw similar results where it's like, well, we can't really tell the difference in the beer. So it's not unreasonable for people to look at the results people got from that and be like, well, you know, it didn't hurt anything. And it seemed like it worked fine. So yeah, maybe I can just use olive oil as a replacement. But as I talk about in the article, there are reasons why that's not really the full story. There was a paper that really, you know, went into meticulous detail and showed really what you had to do to really see the effect of not having oxygen and not supplementing with UFAs and ergosterol. And basically they had to grow cells anaerobically, so no oxygen around, and they would still get growth, a couple of cell divisions, and it would be okay. But then when they'd take those cells and transfer them to fresh media lacking oxygen, growth would cease. So they'd find a point at which growth would no longer occur. So the theory was that there was, as we talked about earlier, you know, an abundance of UFAs and ergosterol in the cells that were transferred to daughter cells, but eventually they run out and can't reproduce any longer.
Right.
And they could take those cells once they'd stopped reproducing and give them ergosterol, and the cells would start growing. They actually didn't even have to give them UFA.
That's the
thing. But in other papers, it had already been established that if you break the gene that the yeast need to make UFAs, you have to supplement UFAs aerobically. So in the presence of oxygen, it seems that you actually need UFAs. But anyway, in this case, you know, anaerobically, you can supplement UFAs and ergosterol. They'll grow just fine. But I think, so this is, I think, explains what we were seeing in a lot of these brewing experiments. You know, people were taking yeast that was healthy and chock full of UFAs and ergosterol and giving it a little bit of UFAs, but they weren't giving ergosterol, but they just did one fermentation. And it's like, yeah, it kind of worked. But I think to really see the deleterious effect of not having oxygen, the best experiments to do from there would have been taking the yeast at the end of that batch and repitching it. And then taking the yeast from that batch and repitching it. At some point, it's going to completely crap out and not make beer. That was kind of what's shown in that Decker article that I talk about.
Gotcha. So, I mean, the initial idea, you know, to utilize olive oil and, you know, try to, I would say, solve the oxidation issues down the line is pretty neat. And they actually did that experiment at New Belgium, like what you mentioned, and the result that they found was not really the answer to them, but it is insignificant in terms of the sensory taste.
Yeah.
And probably people tend to take that to a certain extent, and they can get away with, you know, maybe the first pitch or the next subsequent pitch. But like what you said, you know, if they continuously put yeast through that environment, it will show up for sure.
Eventually you'll have incomplete fermentation, stuck fermentations, bad beer, off flavors.
Right, because the olive oil only supply the UFA to the yeast, but not ergosterol, which has to be either by oxygen or, you know, pure oxygen or sterile air, or the ergosterol supplements that is very expensive, right? Am I getting this right?
Yeah. Okay. So if at least the number I gave at the time, which was, you know, a while ago, I think I came up with around $11 per barrel. So for, you know, per 31 gallons is how much ergosterol you'd have to supplement. So it would all be a very different story if there was a commercially available cheap source of ergosterol. So then maybe we absolutely would. We'd supplement ergosterol, we'd supplement UFAs, and not oxygenate.
No oxygenate at all.
And would probably have some beneficial effects when it came to shelf life. But at this time, there just isn't that, commercially viable source of ergosterol.
Right. Very interesting one. But one thing that I get away from, not get away, but the takeaway for me for this one is oxygenate the word.
Yeah, that's where we are. Until somebody gives us a good source of ergosterol, we're stuck oxygenating.
Right.
I think one of my conclusions too is there's no harm in adding a little bit of olive oil. It's not going to hurt anything, but it's not sufficient. It's not going to supply everything the yeast needs. So you're still going to have to oxygenate.
True. In order to to get a consistent result and make the yeast happy, that's the most efficient way that we can do, and the most simplest way that we can do too. Okay, before we move to the end section of this session, I would just like to go back a little bit at the membrane structure and just quickly point out that the unsaturated fatty acid is the component of the membrane that looks like the tail of the fat content is kind of tilt, just like this, right? Because of the double bond.
Right.
Yeah.
So basically, double bond is kind of rigid. It puts a kink in the chain too. Whereas like a saturated fatty acid would be just a regular,
straight line, right? Yeah. Right. And the ergosterol is the sterol that is the kind of a red figure in the membrane that's kind of randomly stay in between saturated and unsaturated fatty acid, the lipid bilayer, and those are two different things. It's not the same thing.
Right.
And those contribute to a different property of the membrane too.
Right. Yeah. Just completely different compounds that just both happen to require oxygen in their production by the yeast.
Gotcha. I think at this point all of the dots are connected. We understand how vital the oxygen is to the yeast. It for our case, for fermentation wise, it doesn't use oxygen to generate energy. They might do, but that's not the whole point. The important is to synthesize these, UFAs and sterols, and who knows, in the future, like what you said, there might be, viable and commercially sound sterols that they can put. But for now, I guess we are stuck with this oxygenation.
Yep, we're stuck with having oxygen tanks in our brewery, or splashing the fermenter if we're home brewers without oxygen tanks. Yep.
Right. So for this last part, can you walk us through kind of the briefly best practice of how to efficiently oxygenate the wort in the commercial scale?
Yeah, it's definitely with with pure oxygen is the best way to get the amount we need. I think if you're using air and you're pumping air in there for an hour, like the max you can get, I forget, somewhere like 6 to 8 parts per million oxygen. But there's some strains that are really, they want more than that. So 12 to 18 depending on the strain. So the only way you can get to that level is with pure oxygen, bubbled, small bubbles into cool wort is the best way to get to that level. Really, the only way to get to that level.
Got you. So for the home brewers, you can either use the pure O2 injection or rock the carboy, or use the sterile air, like what you mentioned, right?
Yeah, I mean, and that's better than, doing something. Air is better than nothing,
right?
So yeah, so even if that just means splashing or pouring between two vessels to get air in there, that works well too. Once you're at the commercial level, you definitely want to get to dialing in your oxygen at the best level for healthy yeast.
And are there any kind of perfect window to oxygenate? Like, should we oxygenate before pitching or after pitching or in between? Do we have any kind of idea about this?
Yeah, I would say just right before pitching is the best time. So as I think a lot of brewers would do it as they're cooling the wort into like right after the heat exchanger, injecting oxygen through a stone to make sure the bubbles are very small. You get more dissolved oxygen that way. So that's after you cooled your wort is the best, because they can cool wort can hold more oxygen than hot wort. So you don't want to do it, in the kettle or anything like that. You can for very high gravity fermentations, there are times when maybe you want to oxygenate a little bit, maybe 12 hours post pitching yeast to give them kind of a second dose, just because it's a very stressful condition. But I would only reserve that for like very high gravity wort, because you could get far enough along in fermentation with a lower gravity wort that you're just going to enhance staling if you add any oxygen later. Like I said before, the window for brewers is right at the beginning of fermentation, and then do everything you can to avoid adding oxygen. The yeast will be perfectly happy if you add it at any time, but our taste buds won't.
And the line for that high gravity, are we talking about like 20 Plato up for the
that's probably that'd be a good 18 to 20 Plato is a good point to start thinking about doing that second edition.
All right, so just to give them a bit more resource to produce those reserve UFA and sterols.
Yep.
All right. Okay, thanks, Lance. I mean, we are at the end goal of this session. Just to close things out, do you have any final say or thoughts for this topic?
Yeah, I mean, I think I do enjoy that people look into these things. It's a good thing for brewers to be exploring things like that. I just think sometimes brewers miss maybe the larger picture. I mean, yeast is a very well studied organism. I mean, we knew they needed. I didn't discover that they needed oxygen for ergosterol and UFAs. This was known, right? It's just I don't think anybody had put together the whole picture. So we should have already known before we went into it that it wasn't going to work for everything. But still doing the experiment and actually demonstrating it is good. And frankly, I think the first experiment did demonstrate that. I mean, they saw slower growth and they saw differences in the outcome. So right out of the gate, we saw that olive oil was not a suitable replacement for oxygen, and now we know why.
That's cool. Excellent. And I think this is where we will end the session. So Lance, thank you so much for walking us through the role of oxygen in yeast daily life. It is a very delicate topic, and I greatly appreciate all of the inside information that you share with us.
Absolutely. I appreciate the invitation.
Okay. It's time to say goodbye and stop bothering your day now.
My night.
Yeah, your night. Yeah. Thanks again, Lance.
No problem.
All right, man. Yeah, take care.
You too.
Yeah. Here you have it. To this point, I hope that what oxygen is to our beloved yeast becomes more clear or clearer to you all. From my point of view, like apart from zinc, oxygen is one of the essential key nutrition that is not naturally and sufficiently available in the world. And when we subject yeast to that environment every day to ferment, they heavily relies on our interference for them to live happily. And if they do live happily, we will be happy with the beer in our glass as well. So so take good care of your yeast and know your oxygen game. That's all for today. Until next time, cheers and สวัสดีครับ